In this video, I'm going to be talking about a new technology called CRISPR-Cas9. Specifically, I'm going to be addressing how this technology can be utilized to edit the genome of human embryos. So what is CRISPR? CRISPR-Cas9 is a natural system used by prokaryotic organisms to prevent infection by viruses and plasmids. To prevent infection, prokaryotes use CRISPR-Cas9 proteins to recognize and bind to viral DNA and cut it, making it inactive. By using the Cas9 DNA cutting enzyme, a guide RNA that is complementary to the gene of interest, and template DNA, we are able to make cuts in specific locations of DNA, forcing the DNA to repair itself, allowing for mutations, or in the case of editing human embryos, inserting the correct gene sequence. The history of CRISPR starts in 1987 when part of a CRISPR was accidentally cloned instead of a bacterial gene, and the researchers noticed the unusual repeats. In 2000, Francisco Mojica located interrupted palindromic repeats in different archaeal microbes and proposed the term CRISPR to describe them. It wasn't until 2012 when it was proposed that CRISPR-Cas9 could be used for programmable gene editing. So how does CRISPR work? The process starts when a cell is transfected with a CRISPR-Cas9 system. This system includes the Cas9 protein and the guide RNA that matches the targeted gene. Cas9 identifies the corresponding DNA sequence to the guide RNA on the host cell's genome and cuts both strands of the host cell's DNA. There are two repair mechanisms that the cell can then undergo to repair the double-strand break that was created. One of the pathways is called non-homologous end joining pathway. This pathway connects both of the broken ends together and is not suitable for clinical applications as it can cause other mutations and it might not substitute the correct gene. The other pathway is called homology-directed repair, in which a template DNA sequence is used to insert a new gene or correct a mutated gene. This template DNA would have flanking regions that match the target sequence, allowing it to recombine with the host DNA, introducing a replacement gene. A lot of research needs to happen before CRISPR-Cas9 can be used to edit the genome of human embryos. Some current research that is occurring right now is increasing the homology-directed repair pathway over the non-homologous end joining pathway. This can be done by inhibiting key NHEJ molecules. Other research is being done to increase targeting efficiency and stop the creation of mosaics. Mosaics occur when more than one cell in an embryo have different types of genotypes. We also need to determine if mutations could occur at locations other than where we intend to edit. There are 3 billion base pairs in the human genome, and odds are there are locations very similar to the targeted gene. If the guide RNA bound to these areas, we could cause undesirable mutations in other areas. Studies also need to be done on embryo development. We need the embryo to be able to develop normally for clinical applications to be viable. We also need to look at the effects on fertility in the next generation and see how CRISPR-Cas9 affects fertility and see whether the corrected mutation would be able to be passed on to future generations. A study was done on mice with cataracts which showed that fertility was not affected and the corrected allele was able to pass down to the next generation of offspring, but we still need to do some more research on whether these are effective techniques. A study was recently done in August 2017 called the correction of a pathogenic gene mutation in human embryos. The researchers that did this study were able to correct a heterozygous germline mutation in pre-implantation embryos that did not get developed. This mutation is an autosomal dominant mutation that causes hypertrophic cardiomyopathy, which is prevalent in 1 out of every 500 adults. The results of this study showed that by co-injecting the CRISPR-Cas9 system in sperm into M2 phase oocytes, there was more efficient targeting efficiency and there was no mosaic embryos created. They also discovered that the embryos did not repair using exogenous template DNA and rather used the maternal wild-type allele as a repair template. They also found that they did not induce any detectable off-target mutations. The first experiment in this study started out by fertilizing healthy donor oocytes with sperm with a heterozygous mutation. They injected the guide RNA, the Cas9 protein, and the DNA template into the cytoplasm of the zygote during the pronuclear stage, which was 18 hours post-fertilization. They cultured the zygotes for 3 days and then analyzed them by sequencing. The results of this showed that there was a 97.1% survival rate, and the development rates were similar to those of the control embryos. There were 19 control embryos analyzed. Of these embryos, 9 were homozygous wild-type and 10 were heterozygous mutant. This was expected because 50% of gametes inherit a mutated gene. There were 54 S-phase injected embryos. Of the 54 embryos injected, 36 were uniformly homozygous wild-type, 5 were heterozygous mutant, and 13 showed mosaicism. Of the mosaic embryos, there were 67 blastomeres. 35 used the HDR repair pathway, 12 contained a mutant copy, and 20 used the NHEJ pathway. There was no evidence of HDR pathway using the exogenous DNA template, suggesting that homology-directed repair is guided exclusively by the wild-type maternal allele. The researchers then did a second study to see whether injecting during the M phase would result in a higher targeting efficiency and also reduce the amount of mosaics that were formed. For the study, the researchers co-injected healthy wild-type oocytes with sperm that had the heterozygous mutation. They also injected the CRISPR-Cas9 system, including the guide RNA, the Cas9 protein, and a single-strand DNA template. This template contained a known mutation that would differentiate whether the cells repaired using the maternal wild-type allele or the template DNA. 3 days after fertilization, they analyzed each blastomere. Of the 58 embryos that were injected, 16 were heterozygous mutant with non-homologous end-joining repair pathway indel paternal sequences. 42 of the 58 embryos were homozygous wild-type. This means that 100% of the embryos were targeted because the mutation was either repaired correctly, which occurred in the 42 of the 58 embryos, or it was repaired through the non-homologous end-joining pathway and had induced mutations. Of the cells that were corrected to the homozygous wild-type, only 1 embryo showed mosaicism. This was not an issue because the embryo was still homozygous for the wild-type allele. This is because some of the cells in the embryo were repaired using the maternal wild-type allele, and others used the exogenous DNA template, but overall, the genotypes were homozygous for the wild-type allele in all of the cells of that embryo. This study showed that there was no difference between the development of the CRISPR-Cas9 injected embryos and control embryos that were developed into blastocysts. As a summary, the results of this study showed that by co-injecting the CRISPR-Cas9 system and sperm into the M2 phase oocytes, there was more efficient targeting efficiency and no mosaic embryos created. They also discovered that the embryos did not repair using exogenous template DNA and rather used the maternal wild-type allele. They also found that they did not induce any detectable off-target mutations. By non-detectable off-target mutations, they mean that they did not see any changes to DNA sequences that were similar to their target mutation gene. The applications of CRISPR technology are much more extensive than being able to replace a diseased gene in human embryos. This technology can also be used to induce mutations for research animals. It can be used to create genetically modified food and livestock, and we can also use gene drive to quickly spread a mutation through a population. This could be useful, for example, if we were trying to eradicate malaria-carrying mosquitoes. It could also be used to create designer babies and animals by editing the genome to create organisms with more favorable traits. CRISPR-Cas9 is a very powerful technology, and there are many moral and ethical implications that need to be addressed before we move ahead with this new system. Editing genomes can cause serious effects to ecosystems and wild populations, and that could change the course of evolution. There is a lot of effort currently to create guidelines and regulations to this new system, as the effects and power of the system are extreme. Here is a list of the resources that I used in the making of this video and in my research paper. This video is a condensed summary of all the information that was in my research paper. If you would like to learn more please take a look for the paper which will be published alongside this presentation.